Hello and welcome to the first part of our lecture on acellular infectious agents. So we're going to talk about viruses, viroids, and prions. So as always, we start off with a glimpse in history, and the first virus that was ever discovered was the tobacco mosaic virus. And this occurred about in the 1890s, and there were two gentlemen that are credited with this. One is D.M. Ivanovsky, and he was studying the tobacco mosaic virus. And so when plants have this sort of speckled appearance, and even this is true today for plant viruses, that's how it shows up. It shows on their leaves. And so it was ruining tobacco crops. So there was a lot of study focused on that. And at the time, they had filters that were capable of filtering out bacteria. And so he took sap from these diseased tobacco plants, and then he filtered them, and then he took the filtrate, which contained everything but viruses, because viruses are smaller than bacteria and they weren't filtered out. And then he realized that then you could infect another plant. So he called it a filterable pathogen. There was another man, Martinus Bajirnik, that's a fun little name. He went on a couple years after that, and he is the one that actually called it a virus. So from there, it became a filterable virus. In 1915, so 25 years later, there was two men, Twort and de Harrel. One is a Brit, one is a man from France. They were studying bacteria, and bacteria actually get viruses. And when bacteria get viruses, they actually lyse the cells, and you can see these little blank spaces. We're going to find out that those are called plaques a little bit later in this lecture. And so they called them bacteriophages, which means bacteria eaters. Originally, that word virus meant poison. So just a little historical background on our study of viruses. You've seen this slide before. I believe it was in chapter 1. So acellular means without a cell. So we're going to talk a whole lot about viruses, the lion's share of today. Another acellular infectious agent is a viroid. And viroids do not infect humans. They cause diseases in plants. And usually they're going to enter through a wound in a plant. They cause things like cucumber pale fruit and potato spindle. So you see my little messed up potato here. They are similar to a virus in the fact that they are obligate intracellular parasites. They are just a naked, small, circular piece of DNA, and they really don't understand exactly how they cause disease. And that's pretty much all I'm going to say about them, because this is a medical microbiology class. And so we are going to finish off when we're done talking about viruses and bacteriophages, and then we're going to finish off with prions, which we had learned already that these are infectious proteins. So all of these, though, are acellular and are not made up of cells, and they are not technically alive. So first we're going to talk about viruses, and I'm going to first talk about animal viruses because they're of more interest to us. But properties of virus. Number one thing is that they are ubiquitous, and what that word means, that means they are everywhere. They're in nature. They've had a major impact on development of biological life. Actually, 8% of our human genome, so of our DNA, consists of viral DNA. 10 to 20% of bacterial genomes contain viral sequences, and we'll talk about how that occurs. Viruses infect literally everything. Bacteria, protozoa, algae, plants, animals, even viruses have viruses. Yes, that is actually true. It is said that there's 10 million viruses per milliliter of seawater, and somewhere someone did the math, and supposedly that calculates out to 270 times 10 to the 6th, so million metric tons. So there's a whole lot of viruses on the planet. They are inert particles, and what that means, they are not capable of metabolism on their own. They have no enzymes to make ATP or break down nutrients, so they are incapable of metabolism or replication or motility on their own. So they are obligated, so that's what that means, they're obliged, obligate intracellular parasites. They have to live and reproduce inside a host. And how they do that is they're basically going to hijack the host cell's replication machinery. This makes it kind of difficult, so they cannot be grown in pure culture, so we'll talk about 3 methods that are used in the laboratory to grow viruses. But the big thing, the reason it can't be in pure culture is because they have to be grown inside their host cell. And so we looked at this at the very beginning. So they don't consist of cells, they can't metabolize, they can't respond to stimuli. They can multiply, but only inside another living cell, but they can evolve. And we'll talk about how they can evolve and change, basically because they're sloppy. But we'll talk about that in just a little bit. So they're ubiquitous, they're obligate intracellular parasites, and they are structurally compact and economical, meaning there is not a whole lot to their structure at all. They are just genetic information. They either have DNA or RNA contained within a protein coat. So like I said, they are not cells, so they are acellular infectious agents. They are also, if you look at this picture up here, this is showing you an E. coli, and this is showing you a human red blood cell. And then they're showing you the size of the viruses in comparison. So viruses are going to be measured on the nanometer scale. We looked at bacteria in the lab, and they're measured on the micrometer scale. And so most of the viruses are going to fall between 20 and 150 nanometers. What this means is they cannot be seen with a light microscope. So you've never seen one in lab. And it wasn't until the invention of the electron microscope around the 1950s and with special staining techniques that we could actually even see a virus. And so like I said, they were just known as filterable particles. They were crystallized, but no one could actually see them. And just kind of reminding you, micrometers are 10 to the minus 6 meters, and so nanometers are 1,000-fold smaller than that, so 10 to the minus 9. But we measure viruses in nanometers, and they cannot be seen with a light microscope. You have to see them. All the pictures that we'll be looking at in this PowerPoint are electron micrographs. Okay, so just some definitions. Especially in the news, you may have heard, these two terms thrown around. So the virus is that acellular infectious agent. It has nucleic acid and the surrounding protein, and we're going to look at that structure in just a second. You may have heard the term virion, and so when they're talking about virion, what they're really referring to, it's still a virus, but it's a virus in its extracellular state, so when it's not inside its host cell. And so these terms, virus, virion, sometimes people just say the virus, sometimes people are referring to the virion, and when they say virion, they are meaning that it's extracellular, and this is going to be that state that's capable of establishing an infection. So if a virus is inside someone's cell, that virus that's inside the host cell cannot be transferred to another person, but a loose extracellular virion can be. And so that's just the definition of those two terms. All right, so all viruses have these two parts. So the first thing that viruses all have is they have genetic material, and they're going to have either DNA or RNA, but they never have both. So a viral genome is either double-stranded DNA, it can be single-stranded DNA, it can be double-stranded RNA, and it can be single-stranded RNA. It can be in a loop or a circle, or it can be linear. It can be one piece of DNA, or it can be segmented on multiple pieces of DNA. All viruses surrounding their genetic material will also have a protein coat called the capsid. The capsid is going to be made up of identical subunits called capsimeres. If we're talking about the genetic material and the capsid, the term for that is the nucleocapsid. And that kind of makes sense. The nucleic acids and the word capsid is fused. Those viral capsids, and so think of them as that outer shell, inside is going to be the genetic material. Like I said, they're made up of capsimeres, and these are identical protein subunits. What's going to happen inside the host cell, actually, is that this capsid is going to spontaneously self-assemble. So these little capsimeres, so each one of these little balls would be a capsimere, so each one of those little structures is a capsimere. And when it comes time for assembly, which is a stage in the virus replication cycle, they're just going to assemble with very little or no enzymatic activity. And so that's what they mean by self-assembly. There's three different shapes of capsids. One shape is called icosahedral. So icosahedral. So that's this picture up here. Our poster child for an icosahedral virus is adenovirus, which causes respiratory illnesses, so common cold. There are 20 flat triangles. So each one of these triangles looks sort of like a soccer ball, only there's 20 sides to it, and they are triangular. So there's going to be 12 points, and so that's icosahedral. The next one is called helical, and just like a spiral. So helical, the capsomeres are arranged in that helix shape around the genetic information. So it's showing you the nucleic acid, and it's just this circular pattern. The poster child for that one is tobacco mosaic virus, which was the first virus identified. So our next shape is sort of the junk drawer of shapes, if you will. They just call it complex. Bacteriophages, or viruses that infect bacteria, have a complex shape. And this is just showing you a picture. So it's going to have a head that is going to have icosahedral. It's going to be stuffed with the genetic information. These are going to be DNA viruses. And then it's going to have a tail, and then tail fibers for attachment. And so since it was complex, it didn't fit in the other two categories. There are animal viruses like smallpox, which also doesn't fit in the icosahedral or helical category, so it's just called complex. So think of complex like the other, or the junk drawer. So all viruses have a nucleocapsid, so they all have genetic information, and then they all have a capsid. Some viruses, though, when they leave their host, they leave wrapped in that host cell membrane. So these types of viruses right here are called enveloped viruses. You'll also notice there's something else. There's something called matrix protein. You only see matrix protein with enveloped viruses, and it's just extra protein that's going to be between the nucleocapsid and the envelope. It is a structural link between the envelope and then the core nucleocapsid. And sometimes this is going to play a role in viruses that leave through budding. We'll talk about that in a little bit. Some viruses, though, do not leave taking some of their host cell membrane with them. And so these are non-enveloped, or they're called naked, or naked if you prefer. These types of viruses, because they don't have sort of that delicate phospholipid membrane on the outside, these are more resistant to disinfectants. These viruses are going to lead either by lysis or exocytosis, and so we'll look at that in just a little bit. So let's take a look at that outermost part, so the capsid or the envelope. So the outermost part, of course, is going to give that genetic information some protection. This is also the part that is going to be used for recognition. This will be where the recognition sites are. And so we're going to see these spike proteins both on an enveloped virus and on a naked virus. So these are going to be the proteins that are external. These are the proteins that can be recognized by our immune system. These are the proteins that are going to be used in vaccine development. For the coronavirus, SARS coronavirus 2, the vaccine is to give the body a look at these spike proteins so we can make antibodies so we can become immune to them. When you're talking about a bacteriophage, usually those bacteriophages, they're going to attach by their tail fibers. Animal viruses, these spike proteins are usually what they're going to use to dock to their host cells. So we've got DNA, we've got the capsid. Some viruses have an envelope. There are other substances, so in addition to the proteins for the capsid, those envelopes, nucleic acid core, sometimes viruses will also contain enzymes for different specific operations within their host. So it's kind of like a little purse. So they're also carrying other things. So polymerases. We're going to talk about how viruses, whether it's DNA virus or RNA virus, they can use their own viral polymerases. There's a special type of virus called a retrovirus, which has an enzyme maybe you haven't met yet, called reverse transcriptase. We'll look at that. This is how an RNA virus can then create DNA and then insert its genome into its host cell DNA. Proteases are also a viral protein that's going to cleave proteins, viral proteins, once they're made. Some viruses, when they go to assemble, they also sort of borrow, if you will, or package items from their host cell. So there could be some ribosomes packaged from a past host or some transfer RNAs. So there are other things with viruses, but they're basically very simple in structure. Okay, so there's just sort of a pictorial summation of what we just learned. So we know that that capsid can be either icosahedral, helical, or that junk drawer, which just means it has a complex shape. They can be enveloped, like herpes virus, or they can be naked, like the adenovirus, which causes a cold. They can be enveloped, like human influenza virus, or they can be naked, like the tobacco mosaic virus. And then usually if they're complex, this is a bacteriophage. Bacteriophages infect bacteria, so they're never going to be enveloped. They're only going to be naked. So envelope viruses, because that phospholipid bilayer is somewhat fluid, envelope viruses often don't have a clearly defined shape. So often they are termed pleomorphic or variable. So let's take a look at the genetic material. Clearly, there's more variety in genetic material than in cells. The primary way that scientists are going to categorize and classify viruses is going to be based on the type of genetic material that they have. We are used to seeing double-stranded DNA or single-stranded RNA, but there are viruses that are single-stranded DNA viruses and double-stranded RNA viruses. So this is non-existent in any type of cell, so it doesn't exist in plants or animals. As we said before, the genetic material can be linear or it can be circular. It can be a single strand or it can be segmented. It can be anywhere from 4 to hundreds in the hundreds of genes. So just to kind of give you some perspective on how small or how few genes virus travel with, E. coli is about 400 genes and we are about 20,000 genes. So that's just what I mean, but they're very small, economical, and compact. And they're going to rely on their host for most things. So at minimum, though, let's think about it. So a virus has to carry genes for their capsid. They have to carry the genes to copy their genetic material. They also need genes to regulate host cell actions. A lot of viruses are going to shut down protein production by their host cell. And then they're also going to need proteins for packaging of their mature virus. So classification of viruses. There is actually a committee. It's called the International Committee on Taxonomy of Viruses. And it's their job to basically name and organize all of the viruses. And so they've kind of had, like everything else in biology, sort of a restructuring, if you will. And we're not going to learn too much about the classification of viruses, but viruses are placed within families, and families are going to always end in the suffix viridae. The names don't really follow a consistent pattern, but a lot of times they're going to be named and sort of sound like what their disease is. Sometimes they're named for how they look. So coronaviridae, the corona means crown, and yes, this is SARS right here. And when they looked at the electron micrograph, they thought it looked like a crown. Other types of viruses, like Bunyaviridae, is named from where it first originated. So Bunyamwera, that was something I had to practice. But there really isn't a really method to the madness, if you will. So let's just kind of take a look. So they do always classify them DNA viruses or RNA viruses. I'm going to show you kind of how I want you to learn them, and I'm going to give you some generalities about DNA viruses and RNA viruses, but I do not expect you to memorize this table. But just kind of take a look. We're going to talk about the human papillomavirus, which causes both genital warts and other warts, and it also is a virus that can cause cancers later in life. But you can kind of look at their family name, so Papillomaviridae, and so that kind of makes sense. I kind of talked to you about adenoviruses. They cause respiratory infections. A lot of those are just going to be cold, upper respiratory infections. So they come from Adenoviridae. Herpes viruses come from herpes viridae. So a lot of times it does kind of go together. There are some cases where, like the polio virus that causes polio, the long name for that is poliomyelitis, is in the family Picornaviridae. So, which also hepatitis belongs to that group. So the naming, I just thought for our purposes, we're spending one day on this. That isn't very helpful. So clinicians kind of, and that's just kind of showing you single-stranded DNA, and then we're going to have special retroviruses, which we'll talk about. And so I'm going to talk about all the different types of viruses, but we're not going to really focus on what family the virus went to. So this is kind of what I want you to know. So clinicians, they're going to use the vernacular names or the short names for virus, and then usually that's going to be the species name, but not always. And I think it's more helpful if you kind of categorize some of these as opposed to what body system they affect. And so let's go ahead. We're going to start over here with respiratory viruses. When someone has a respiratory virus, either it's going to be an upper respiratory virus, so just sort of affect the nose and the throat. So we're talking about runny nose, itchy, scratchy, red throat, maybe sort of a loss of voice. That would be an upper respiratory infection. When it infects the airways and the lungs, that's when it's a lower respiratory infection. That's when you can get pneumonia or ARDS, acute respiratory distress syndrome. And some of the really common ones, hey, look, SARS, coronavirus. You probably knew that one already. Rhinovirus is really causes the majority of respiratory illnesses. Adenovirus is another one. Parainfluenza virus, influenza A and B, and then RSV, it has respiratory in the name. We're going to talk about this one a little bit as well. When it comes to stomach viruses, so gastroenteritis, most of these are going to cause either vomiting and then diarrhea, or both at the same time. But these are going to be transferred through the fecal-oral route, respiratory. It's going to be through respiratory droplets. And norovirus is a really common one. If you hear about, outbreaks, sometimes it happens on cruise ships or in schools. Norovirus is a common cause for those. Rotavirus, this can cause diarrhea, usually causes diarrhea in young children. There is a vaccine for it, so not a super large problem in the US. Sexually transmitted viruses, so herpes, human papillomavirus, which is genital warts. We're going to talk about HIV, the virus that causes AIDS, but those are all going to be transferred by sexual contact. When we talk about skin infections, or viral skin infections, we're talking about ones that cause rashes, basically. We're going to talk again about varicella zoster, which is the virus that causes chickenpox, and then later in life can cause shingles. Herpes simplex type 1 is the type that causes cold sores. We'll talk about that because that is another latent infection. Smallpox, which has pretty much been eradicated in the US since 1972. I believe they declared it eradicated worldwide, I think it was 1980s. That one, once again, is another one infecting the skin. There's something called fist disease. This is also called slap cheek syndrome. This is another viral infection of the skin. And then measles, rubella, which is German measles, so MMR vaccine is kind of taking care of those for us as well. And then warts, so just common warts, are caused by a human papillomavirus. So these are all viral infections. Hepatitis means inflammation of the liver. And so there's different types, A, B, C, D, and E. And some of these are different in how long they last, different parts of the world. Hepatitis A is usually an acute form of liver disease, so liver infection usually resolves itself. B and C, B is going to be our poster child for a chronic viral infection, so we're going to look at that in a little while. B and C can also cause liver cancer later in life. So we're going to talk about those. D and E are not very common in the US. Mumps is a disease you may have heard about. It is caused by a virus of the salivary glands. So it infects the parotid salivary gland and causes inflammation. And then lastly, up here, encephalitis, so affecting the central nervous system. We've talked about meningitis. viruses that affect the central nervous system. So rabies, so this is a disease that you would get from either being bitten or scratched by a rabid animal. Without treatment, it is fatal. Once again, because we vaccinate our pets, it's not a very big player in the US, but worldwide, because there's that lack of vaccination. Still, there are a lot of rabid dogs. It is a problem in other parts of the world. Arboviruses, these are arthropod transferred viruses. The one we're going to talk about is West Nile virus. That is the big one that can cause encephalitis. And then enteroviruses, we're going to talk about polio and how polio moves, but from one location to another. So we'll mention that one in a little bit. But just more, I put it in your Quizlet for you, just sort of in groups, and I don't expect you to know every detail about these viruses. Okay, and so I just kind of threw this slide in here, and this is why I picked the ones that I did. It's showing you enteric viruses, respiratory viruses, sexually transmitted viruses, and zoonotic. Remember, that term just means it's from animals to humans, and so either through an arthropod vector or from the animal directly in the case of rabies. We don't really have a whole lot of cowpox going on anymore, but that too would qualify. And just kind of showing you, this is several of the respiratory flus. The number one's going to be the rhinovirus. So clinicians kind of know more which viruses affect what system. So that's kind of how I thought we would approach that one. Okay, so the next thing we're going to look at is viral replication. And I've kept the same steps whether we're talking about a phage, so a virus affecting a bacteria, or an animal virus. And so we're going to look at attachment. So attachment is attachment of that virion, that extracellular virus particle, to its host cell. Then we have to have entry of that virion. The genome has to get into that host cell. So we'll look at that process. Then it's the virus's job, basically, since the parasite is to reproduce. So we're going to get synthesis of new nucleic acids and viral proteins. And this is when that virus particle is going to hijack that host cell. Then we're going to get assembly of the new virions within the host cell. So we have to get that genetic material packaged inside that nucleocapsid. Any sort of spike proteins need to be assembled. And then that last step is going to be release of those new virions from that host cell. So let's go ahead and take a look.
You probably already know that viruses are responsible for causing a number of diseases, from AIDS to the flu. But did you ever wonder how a virus works? Viruses are basically infectious particles that take over the operation of a cell for the sole purpose of manufacturing new viruses. How exactly does a virus do this? In many ways, it works in the same manner as a computer virus does. It has to first gain access and then convince the machinery within the device, in this case a cell, to make multiple new copies of the virus. There are many different types of viruses, but they do share some similar characteristics. First of all, viruses are usually specific in the types of cells that they infect. The specificity of a virus is dependent upon the types of receptors that are found on the surface of the target cell. Every cell in your body has a pattern of protein receptors on its surface. The virus uses these proteins to target specific cells for infection. In this example, the genetic material of the virus infecting the cell is DNA. However, unlike the complex DNA found in the nucleus of a cell, the DNA of a virus is relatively simple and just contains the information needed to manufacture new virus parts. Once inside the cell, the instructions in the DNA are transcribed to RNA. The protein-building machinery of the host cell then translates these instructions into the components of a new virus. These parts are then assembled into new viruses within the host cell. When ready, they emerge from the host cell, often killing it in the process. As they emerge, some viruses retain parts of the host cell membrane, forming an envelope around the virus. This envelope gives some protection to the virus from the immune system of the host organism. Each new virus is now capable of infecting another host cell and repeating the process of virus replication. By understanding how the life cycle of a virus works, scientists have been able to develop antiviral drugs that target specific points in the virus life cycle and thus prevent the virus from replicating.
Let's look at some details of viral replication. And this is going to be viral replication in an animal cell. So that first step is attachment. Some books call that step adsorption. I wanted to keep the steps the same for both animal viruses and phages. So that is when the virus is always going to have this surface protein that is going to bind to a receptor on its host cell. Usually it's going to be a glycoprotein on that cytoplasmic membrane, and usually it's going to be that spike protein. Some viruses have more than one receptor for attachment. For example, HIV, the virus that causes AIDS, there has a receptor, a main receptor, which is the CD4 marker, which is on helper T cells and other white blood cells like macrophages or dendritic cells, and it also has a co-receptor called CCR5. The thing about these receptors that I kind of want you to know is that these receptors, it's not like the host cells, it's not like we have cells in our body that put out targets for these viruses intentionally. All of these proteins have a normal function within the host cell that's unrelated to viral infection. So SARS-CoV-2, so SARS coronavirus 2, its receptor is going to be the angiotensin converting enzyme 2. And so this is going to be found on multiple types of cells. So let's kind of look at this for a moment. So we're talking about attachment. So the fact that there's sort of this viral host cell attachment, it's going to be very close, so it's going to fit like a lock and a key. What this does is it determines two things. It determines tropism and the host range for that virus. Tropism refers to the cell type or tissue that a particular virus can affect. And so that ACE2 receptor for SARS-CoV-2, this receptor is found on epithelial cells of the respiratory tract, both upper and lower. It's found on arterial cells, it's found on cells of the gastrointestinal tract, and it's also found on the kidney. The difference between SARS-CoV-1 and SARS-CoV-2, there was a deletion in the RNA, so in the viral genome, and that sort of expanded tropism. SARS-CoV-2 mostly infected lower respiratory, whereas SARS-CoV-1 also infects the back of the throat. That's why the test was those nasopharyngeal swabs. The receptor on the host cell is also going to determine species type, and so that's called host range. Most viruses just infect a single species, like dogs don't contract measles from us, but that's not always true. So like the rabies virus has a wide range, so it has a wide host range. You can infect dogs, humans, and also some other wild animals. Coronaviruses also have a wide range, so they can infect bats, swine, pangolins, civets, and mice. And so the current virus, so SARS-CoV-2, it is very similar to a virus that was in bats. And so trying to figure out how do you go from bats to humans is because it has a wide host range. It could be that someone caught it from a cave, or more likely there was an intermediate host. But that attachment between virus and host cell determines its tropism, its tissue type, so what type of disease can it cause, and then also the host range. Can these viruses go between species? That all has to do to the similarity of those receptors. So let's look at entry.
There are two mechanisms by which enveloped viruses enter host cells. In one of the mechanisms, the virion attaches to host cell receptors by specific proteins on its surface called spikes. The envelope of the virus fuses with the plasma membrane of the host, and the nucleocapsid is released directly into the cytoplasm. The nucleic acid then separates from the protein coat. In the second mechanism, the enveloped virus adsorbs to the host cell by specific proteins on its surface, and the virion is taken in by endocytosis. In this process, the host cell plasma membrane surrounds the whole virion and forms a vesicle. The envelope of the virion then fuses with the plasma membrane of the vesicle, and the nucleocapsid is released into the host cytoplasm. The capsid protein is then removed, releasing the nucleic acid of the virus. A naked virion also enters by endocytosis. Since the virus has no envelope, it cannot fuse with the plasma membrane. After being engulfed, the viral nucleic acid is released from the endocytic vesicle. The nucleic acid then separates from the capsid.
All right, so entry. So we have attachment, entry. So there's two parts to entry. There's penetration and encoding. So penetration can occur those two ways. We just watched it. One is membrane fusion, and it can only be enveloped viruses that are going to enter this way. This is the way SARS-CoV-2 enters. Another way to enter, which can be either for an enveloped virus or a naked virus, is through receptor-mediated endocytosis. And so the surface proteins, those spike proteins, are going to dock with the receptor on the host cell, and then that's going to be brought in through endocytosis. Once the virus is in, the next step is that uncoding, and that term for uncoding just means the nucleic acid is going to separate from that protein coat, that capsid. The next step is going to be synthesis, and so there's two parts to synthesis as well. We're going to have expression of viral genes, and this just means protein synthesis. So the capsid proteins, any sort of enzymes required for replication or to take over the host cell. Often, these are going to be synthesized as one long protein, and so it's a polyprotein. And then it's going to be a viral protease that will then cleave that large protein into the functional proteins. And since this is going to be a viral protein, this is a common target for antiviral medication. So there are antiviral meds that target the HIV protease. There's antiviral meds that target the hepatitis C protease. So that is a really common site of antiviral medications. And then, of course, the next thing that has to occur is that replication of the viral genome.
This is the translation event whereby a host cell ribosome will move along a molecule of messenger RNA, and the two of these in tandem are involved in the production of viral proteins. These viral proteins, in fact, include the viral enzymes that are synthesized in the form of long precursor proteins. Different types of messenger RNA molecules, in fact, exist in regard to the virus, and these are translated into different types of viral proteins or polypeptide chains. This is the protein cleavage event, whereby the protease enzyme of the virus cuts these long, non-functional polypeptide proteins into smaller individual subunits that indeed are fully functional. This is an essential step in virus replication. The protease inhibitor, or PI, will now attempt to block the function of the protease enzyme. This is the binding of the protease inhibitor to the protease enzyme, an event which is essential in blocking the ability of the protease enzyme to function, and this then results in preventing the protease enzyme from being able to cleave the large non-functional polyprotein precursors into their final functional units. Hence, if we succeed in blocking the protease enzyme, we will simultaneously prevent the virus from being able to fully assemble in the context of new, fully infectious virus particles. The blocked protease enzyme is now unable to cut the long polypeptide chains into the smaller individual subunit proteins. Hence, the protease inhibitor, or PI, will block the process of protein cleavage and prevent the formation of new viruses. This then results in the arrest of virus replication and clearly is a step that is essential in preventing the formation of new infectious virus, and obviously is a step that has been successfully targeted through the development of new inhibitors of HIV replication that is essential to the success that we have achieved in the therapy of HIV disease.
So your book goes into quite a bit about the synthesis of the viral genes. The big thing I kind of want to focus in on is that regardless if it's a DNA virus, double-stranded, single-stranded, RNA virus, positive sense or negative sense, all viruses have to get to what's called that positive sense RNA. That positive sense RNA acts just like messenger RNA, meaning it can be read by the host's ribosomes and translated into viral proteins. And so these are all just mechanisms in order to get to that positive sense RNA that can then make those viral proteins. And then that second part of viral synthesis is then also that genome has to be copied. And we talked about genetics. You know that there's complementarity between the bases, so A, T, G, C, or AU if we're talking about RNA. And so whatever viral genetic material that virus comes with, it is going to copy itself using that complementarity. And so viruses, that's one of the ways that they evolve, because when they copy their genome, they're usually using their own enzymes and they're sloppy. So I'm not going to ask you this part. I kind of summarized it in a table because it's kind of more the take home, the practical message, if you will. And so I've kind of made some generalizations. So most DNA viruses, the majority of them are going to be double-stranded. The majority of them, of those double-stranded DNA viruses, are going to be icosahedral. They are going to get to that messenger RNA, since they're rDNA, just by using host cell RNA polymerase. To copy their genetic material for replication, they can either use the host cell DNA polymerase, or they can carry their own. Usually, this is going to occur. So their DNA. So they're going to go to the nucleus. They're going to get, that's where the RNA polymerase is. They're going to get transcribed, and then we're going to get protein synthesis. Just some examples of DNA viruses: smallpox, herpes, papillomavirus, adenovirus, chickenpox, that fifth disease, that flat cheek syndrome. This is going to be single-stranded DNA. These are all double-stranded DNA. I'm going to bring up before how herpes and the papillomaviruses, these are going to be linked to cancer later on. So I'm just going to tell you most of the viruses that are linked to cancer or tumorigenic viruses are DNA viruses. Pop over here because we're kind of camp out here for a little bit. Most RNA viruses, this is generalizations once again, they can be either positive stranded, which means they can immediately act as a messenger RNA, or negative stranded, antisense. So then they have to be transcribed, so you have to go use this negative strand as a template to get positive strand that can then be converted into messenger RNA. That is usually done by what's called an RNA-dependent RNA polymerase. Your book collectively called that replicase. The thing I'm pointing out and why it's in blue is that this particular enzyme that's necessary to make viral proteins and to copy the RNA is a viral replicase. And viral enzymes that copy DNA, copy RNA, they lack proofreading ability. Our DNA polymerase is a proofreader, so it can back up and correct mistakes. Most viral polymerases do not have proofreading capability, which makes them sloppy. And this is what's going to lead to their evolution or their change. We're going to look at antigenic variation, antigenic drift in just a minute, is because viruses are sloppy. Since RNA belongs in the cytoplasm, that's where it's going to be translated. Usually those viruses can replicate themselves and make their proteins in the cytoplasm. We're going to look a whole lot about coronavirus. Poliovirus, influenza virus is an RNA virus. Rhinovirus that causes colds, that's why you're never really immune to a cold, is also an RNA virus. Double-stranded RNA, reoviruses, that's a family. We talked about the rotavirus that causes diarrhea disease, usually in children. Retroviruses, these are single-stranded RNA. They usually have a complex shape. And what's special about a retrovirus, retro means backwards. Retroviruses are RNA viruses that carry an enzyme reverse transcriptase so that they can go backwards. They can go from RNA to DNA. And so they come carrying this enzyme, and once again, it's a sloppy enzyme. We're going to look at HIV, and that's why there's a lot of variability in HIV. They are going to make their messenger RNA by using usually the cellular RNA polymerase. It's going to occur in the cytoplasm, and like I said, our hallmark for retrovirus is HIV. So, looking specifically at RNA viruses, and we are looking at, your book uses influenza, which is always just the poster child for what's called antigenic drift and antigenic shift. But this can occur in other viruses as well. So an RNA virus requires an RNA-dependent RNA polymerase because we don't go from RNA to RNA in our cells. So this is going to lack proofreading ability. So it makes it sloppy when it copies, so it makes mistakes. And so this is going to generate mutations. And so what you get is called antigenic drift. So this is why there is season-to-season chains of certain viruses, or why you can't gain immunity to the flu, like why you can get the flu more than once, you can get colds more than once, because one virus can just slightly change itself through mutation, and it just looks a little different, which makes it new enough to your immune system that you are not immune. Some viruses have segmented genomes, meaning their genome is on multiple pieces of RNA. So this is the case with influenza, but there's other RNA viruses as well. So what can happen in this case is that you've got influenza virus A, influenza virus B, and they happen to infect the same host, which really is not all that uncommon. When viruses go to assemble themselves, so put themselves together, they get kind of sloppy packaging. Instead of just grabbing their own genes, they grab genes from another virus. And so that's called reassortment. And so this sort of reassortment process is how you end up with what's called an antigenic shift. And so I always kind of remember the difference because drifts seem smaller than shifts. So drifts are small little changes, so season to season differences, and shifts are large changes. This is the kind that leads to novel viruses and pandemic influenzas. So let's go ahead and watch.
Antigenic Shift and the H1N1 influenza A virus. Antigenic shift occurs when the influenza virus goes through the major antigenic changes as a result of the reassortment of genomes among different strains, including animals. Origins of the hemagglutinin HA, nucleoprotein NP, and non-structural NS gene segments of the H1N1 virus are linked to avian flu virus, which infected pig populations around 1918. Let's review the principles of antigenic shift. Influenza sets up local upper respiratory tract infections in pigs. The virus targets and infects epithelial respiratory mucosa cells. Hybrid viruses are created when they infect the same cell by different strains of the virus. First, the native pig strain of influenza A targets the epithelial cell. It binds and enters the host cell through endocytosis and releases its viral genome into the cytoplasm. Secondly, a bird strain of influenza virus A infects the same cell and mixes with the already present pig strain gene segments. Mixed viral gene segments from bird and pig strains reassort into a new influenza A virus strain, which may further spread to the human population. Another major antigenic shift of influenza A took place around 1979. The neuraminidase or NA and matrix MA gene segments originated from wholly avian influenza viruses and entered the swine population. Substantial antigenic shift of viruses have occurred since 1979. For example, when the polymerase acidic protein or PA and polymerase basic protein 2 PB2 segments from the bird strain reassorted into a new strain, which entered swine in North America around 1998. Around the same time, the polymerase basic protein 1 PB1 gene segment migrated to a swine strain from humans. This had originated from birds around 1968. Evolution via the combination of various antigenic shifts resulted in the novel H1N1 transmitted to humans in 2009.
Okay, so that's what we just watched, and this is a picture from your textbook. So think drifts are smaller than shifts, and so antigenic drifts are responsible for seasonal influenza changes, such as slight mutations, and then antigenic shifts, which is like we saw, that's what's involved in the pandemic influenza. And that's when you have that reassortment, when you get one host that's been infected with two different types of influenza. Okay, so we are going through our replication. And so we had attachment, we had entry, we had synthesis, and now we need to put the parts together, and that's called assembly. Assembly and maturation. And like I said, this is spontaneous self-assembly, and that just means it doesn't involve a whole lot of enzymes. It does vary as to where this is going to occur in the cell. So if it's a DNA virus, usually this occurs in the nucleus. If it's an RNA virus, it usually occurs in the cytoplasm. And this is just kind of showing you a picture of the helical capsid, icosahedral capsid. And then there's some special ones, like in a bacteriophage, that's going to require some extra scaffolding proteins. But for the most part, what I want you to know is that it's just pretty much spontaneous. The last step in viral replication, so hang with me, the last one is release. So these release of new virions from the host cell. And so that can occur in three ways. So enveloped viruses, the only way you can get an envelope is through budding. And so the spike proteins, as they're being made, they're going to be inserted into the host cell membrane. And then the nucleocapsid is going to come close to that membrane. You've got matrix proteins there. And then the nucleocapsid is going to be extruded from the host cell, becoming coated with the host cell membrane that has the spike proteins. And then that new virus is released. Notice that there's this pinching off from the cell membrane. This does not kill that host cell right away. So this cell can continue to release or shed virus particles. Kind of hold on to that thought. The other two ways, so we've had budding. One other way is through exocytosis, and so that's when that virus particle is ejected through a vesicle, so coming from the endoplasmic reticulum to the Golgi apparatus and then being released. And then the other way is going to be through host cell lysis. So that's actually the rupturing of the host cell. Often this is going to be through apoptosis, which can be initiated either by the virus or the host cell. When a cell is lysed, this is when death occurs. So budding and exocytosis doesn't kill the cell right away, but lysis does. All right, so think about it. So now we have a host cell that was infected, the virus particles got released, so what can happen next? These virus particles can go off and infect a neighboring cell, or they can travel through the bloodstream, or if they're in tissues, they can go to the lymphatic system, and then they can travel to other parts of the body. So poliovirus, initially that is a respiratory infection, so it infects the throat. It can also infect the intestinal tract. But then it's going to enter the bloodstream, and then it can spread. And so if it spreads and attacks motor nerves, that's when you get the paralysis of polio. Maybe you've heard of the March of Dimes. Like I said, we've kind of eradicated this through vaccination, which is awesome in our country. Or it can also spread to the central nervous system and cause meningitis. So infecting neighboring cells or travel to infect other organ systems is what occurs when a virus is released. And this, of course, is assuming the immune system doesn't get a hold of it and eliminate it first. How do viruses do damage? So think about it. They are infecting host cells. They're taking that cell over, so usually it's going to be interrupting function. And then two, if that virus particle leaves through lysis, that is going to kill that host cell. And so if there's a lot of lysis and a lot of cell death, that's going to take some recovery time. But the majority of the damage done by a virus is going to be through collateral damage. Most viral diseases sort of follow the same pattern, which usually there's a prodromal phase where someone just sort of feels kind of sick. So just kind of like, oh, I feel a little run down, I'm a little bit achy, and maybe a little tired. But then it's going to go into full-blown fever and aches. Usually this is going to be because of the collateral damage, the immune system trying to clear the virus, and then you can also get that cytokine storm. And so that overproduction of cytokines. So this is going to lead to an exaggerated immune response that causes damage. We're going to look at, in the next lecture, how sometimes too that immune response can even get to the critical level. Okay, we're going to stop here. Hope you enjoyed it. Have a great day.